Hello and welcome to the online classes by Saket Girls PG College. I am Dr. Deepak Chandra Saroj, an assistant professor at this college. So today we will be discussing about the molecular structure of the DNA, and this is also known as the blueprint of life because all the information needed for a particular cell or life is imprinted into the DNA. Let's see about more about it. So what is DNA? So DNA is genetic material of all living organisms, and also in the DNA viruses it is the genome. Except RNA viruses, it is genome for the all living organisms as well as the DNA viruses. And what it does, it carries the genetic instruction for the development, cellular growth, reproduction, etc. And structurally it is composed of two long polynucleotide chains which are like twisted ladder. So in this lecture, in this presentation, we will be seeing the detailed molecular structure of the DNA, what are the components, how DNA is linked and formed. So DNA has major three components. One is the sugar component, which is present in the form of deoxyribose sugar, which is 5-membered carbon ring compound that is called pentose sugar, and it forms the backbone of the DNA. And second major component is the nitrogenous bases. We have two categories of nitrogenous bases, one is purine and another is pyrimidines, and this form the rungs of the DNA ladder. We will see all these things in detail in upcoming slides. And third group is the acidic group, that is the phosphate group. It comes from the phosphoric acid and it gives the negative charge to the DNA molecule. DNA molecule carries net negative charge and it comes from the phosphate group. We know phosphate has 3 negative charge. So, and it connects the nucleotides. We will see what are the nucleotides in upcoming slides, and the phosphoric acid, this phosphate is the linking agent between the different nucleotides. So first component is the sugar component that is made up of deoxyribose. What is deoxyribose? Its basic component is the ribose sugar, which has 5 carbon, 1, 2, 3, 4 and 5th is outside. So this ring in which there are 4 carbon and 1 oxygen, this type of ring is known as Furanose ring, and at the second position, at each carbon we can see there is one hydrogen and another hydroxyl group, but at the second position in the ribose there is hydroxyl group, but when this hydroxyl group is replaced by hydrogen at second carbon, then it is called as 2-deoxyribose. So if we see the chemical formula of this ribose, then it will come as C5S10 and O5. C5S10 and O5, and here there will be S9 because O4. So what is the role of this? When this is deoxyribose, this is more stable than the ribose. Therefore, DNA molecule is more stable than the RNA molecule. And second component is the nitrogenous base. And we know that there are two groups of nitrogenous bases. One is the purine. In the purine, it is made up of 2 ring structure. One is the pyrimidine ring and this is the second one is the imidazole ring. So together these form the purine rings, and there are 4 carbons in the ring. At first position 1, 3, 7 and 9th position we have nitrogen, and similarly here also at 4 position in the ring we have nitrogen, and the Two types of purine bases are found in the DNA. One is adenine, which is known as 6-amino purine. At 6th position we have amino group here, while in the guanine we have at 2nd position we have amino group, and at the 6th position we have oxygen. So this is known as 2-amino 6-oxypurine. And second group of the nitrogenous bases are known as pyrimidine nitrogenous bases. There are two types of pyrimidine nitrogenous bases found in DNA molecule, and one is cytosine and another is thymine. The cytosine has at the 2nd position oxygen and the 4th position amino group. So its name is 2-oxy-4-amino pyrimidine. This is its chemical name. And in the thymine we have at 2 position we have oxygen, that is 2nd and 4th position, and at 5th position we have methyl group. So its name is 2,4-dioxy-5-methyl-pyrimidine. We will see how these are interlinked and forms the DNA in upcoming slides. So third major component of the DNA is the acidic component, that is phosphate, which comes from the phosphoric acid H3PO4, and at physiological pH this phosphate exists as phosphoric acid is ionized and in the form of phosphate and protons. So, and this phosphate gives net negative charge to the DNA molecule and it helps in the polymerization of DNA. We will see in further slide. So, when we are talking about we are discussing about the assembly, how these three components combine together to form the monomeric unit of the DNA. So first step is the formation of nucleoside, where nitrogenous base is connected to the sugar compound, the deoxyribose compound. At first carbon, this is the first carbon of the deoxyribose, and it connects with the nitrogenous base, nitrogen of the nitrogenous base, and this bond is known as beta N glycosidic bond. And we have 4 types of nitrogenous bases in the DNA. Therefore, there are 4 types of nucleosides in DNA based on the nitrogenous bases. If adenine is connected with deoxyribose, then it forms a deoxyadenosine. Similarly, if guanine is attached with deoxyribose, then it forms deoxyguanosine. Similarly, others. And next step in the assembly of these 3 components is the formation of nucleotide. Nucleotide is formed when nucleoside, as we see earlier, nucleoside is attached with phosphate group, then the resultant structure is called nucleotide, which is this nucleotide serves as the monomer of DNA molecule. So this is the building block of DNA molecule. And so here this structure, this is nitrogenous base, this is sugar, and together this much structure is known as nucleoside. So this bond is beta glycosidic bond, and this is the 5th carbon. This is the 5th carbon of the sugar, the deoxyribose sugar, and 5th carbon is connected with the oxygen of the phosphate group. So in this way, this is the ester bond, because this is the acid and this is the hydroxyl group, which is in compared to this, that is basic. So therefore it is a ester bond, because in chemistry, if you remember, we have carboxylic group COOH and alcoholic group and when they both react, they form the ester bond. Similarly, here we have a special type of the acid. It is not carboxylic acid. Therefore, this is the ester bond which is phosphoester bond. And now this complete structure is known as nucleotide, as we have 4 types of nitrogenous bases in the DNA. Similarly, based on those nitrogenous bases, we have 4 types of nucleotides as well. And how these nucleotides are the monomer of DNA molecule, and how these are interconnected, and after interconnecting they forms a long chain of these nucleotides, that is called as polynucleotide. So how this polynucleotide gets formed, let us understand. So this is the nitrogenous base, this is the deoxyribose sugar, and this is the phosphate, and this component is called as nucleotide. And similarly, here we have another nitrogenous base, another sugar, and another phosphate group. So this is another nucleotide, and both nucleotides are interconnected. So this phosphate of this nucleotide is connected at the third position of the deoxyribose sugar. This is the third carbon. With the third carbon, this phosphate gets connected. So at this position, at the first nucleotide, this phosphate is present with the fifth carbon of this deoxyribose sugar, and this is the third carbon. So in this way, this phosphate group helps in interconnecting the nucleotides, successive nucleotides. In this way, we have one ester bond here and one ester bond here. Therefore, this is phosphodiester bond, both bonds. So this phosphodiester bonds helps to interconnect the successive nucleotides. In this way, nucleotides keeps on connecting and forms a long polynucleotide chain, which you can see here. And at the one end, we have, it also establishes the directionality of the chain. It has free 5' phosphate and 3 hydroxyl group. When this now, like we have seen here, this is the phosphate group, it is free here, and this is the third position of the deoxyribose, which this hydroxyl position is free here for upcoming nucleotides. So this end of the polynucleotide chain is known as 3' end, and this end will be known as 5' end of the polynucleotide. Now let us understand how DNA structure actually looks like in X-ray, which was solved, X-ray structure was solved by the Watson Crick, and they proposed this model of the DNA molecule, which is known as the double helix DNA molecule. It has 2 anti-parallel strands, mean one strand, we can say this is one strand and this is another strand, and this end is known as 5' end. Here you can see the labeling, and this another end of the another polynucleotide chain is 3' end. So these both polynucleotide chain are anti-parallel, and this helix is right-handed helix, and Chargaff gave how these nucleotides, the nitrogenous bases of the nucleotides, are interconnected with hydrogen bonds, and he suggested that he gave that adenine connects with thymine with 2 hydrogen bonds, and similarly guanine connects with the cytosine with 3 hydrogen bonds. And when this polynucleotide is twisted in a ladder form, as we can see here, it forms this group. This is the bigger group and this is the smaller compared to this. So this is the major group, this is known as major group, and this is the narrow group, and these groups are very important for the DNA in order to function it, because these groups helps proteins and enzymes to get attached and decode this DNA or replicate this DNA as it is required in the DNA, because DNA is the molecule, it has the blueprint of all the reactions and functions cells carries. So if DNA polymerase and ribo and when it has to transcribe, then all the components, all the machineries for polymerization and transcription are here. They comes and binds to the major groups. Thus major and minor groups, the helical twist and backbone spacing creates grooves that are essential for protein binding. And this is the pitch of the DNA, that is 34, because one twist, this is one helix from here to here, it takes that is the 34 angstrom distances, and 3.4 angstrom is the distance between the successive nitrogenous bases. So in summary, DNA is the blueprint of life. It is made up of three major components. One is sugar component, that is 2-deoxyribose, and another is the nitrogenous bases. We saw that there are 4 types of nitrogenous bases: adenine, thymine, guanine, and cytosine. And third component is the acidic component, that is phosphate, which comes from the phosphoric acid. And this sugar, the deoxyribose, gets connected with the nitrogenous base and forms the nucleoside, and further this nucleoside interacts with the phosphate and forms the nucleotide, and further these nucleotides polymerize and form the complete DNA molecule. Thank you so much.